Very good morning. So in this particular lecture schedule, that information will be provided to you about seed development maturation, particularly in the case of angiosperm, that to sexual mode of reproduction. So this is an overview with reference to that megasporogenesis and megagametogenesis. Before entering into that megasporogenesis, I want to give you some information about that microsporogenesis also. Here you can see, so this is a filament, so that will have a anther sacs will be available. Within that anther sac, that will have a pollen grains. So pollen grains are the male gametobite that is available in each and every that pollen that have two male nuclei. So that pollen grains will be released at the time of maturation of that anther sac. So the anther sac will burst and release the pollen grain. That pollen grain will reach us that stigma. So this is a female, that entire thing is a female organ. So this particular part is a stigma where that pollen grain will land. So these are things happening at the time of that fertilization process. So before getting into that fertilization process, I want to give you some information regarding that megasporogenesis. So this is a female starting from that stigma to that ovary. That is that female organ in the case of a plant system. So this particular part is the ovary. Within that ovary, then two dots is there. So both of them are ovule. So the ovule tissues, that is a megaspore mother cells, deploy in nature, that will undergo that meiosis process. Through that meiosis, the cells undergo reduction divisions. That leads to formation of the 4 haploid cells. So 4 haploid cells will be developed. Out of 4 haploid cells, 3 cells were degenerated, 1 cell alone will survive. That survived cell will undergo that mitosis, that is a multiplication that attain to cell stage. Then further round of cell division, that will form a 4 cell stage. And then a 8 cell stage that will reaches. After that, the cytoplasmic division will be occurred, that leads to formation of the matured embryo sac. So this is a mature embryo sac. That the embryo sac consists of the 8 nuclei. 8 nuclei here, that 3 nuclei, then 2 nuclei, another 3 nuclei. So it's a 8 nuclei structure, but if you consider in the name of cell, that is a 7 celled structure, because these 3 cells, these 3 cells normally known as antipodal cells, and these two are 2 nuclei, but with reference cell it's a single cell. So single cell structure. Then this is an egg cell, on either side 2 antipodal cells. So this is a mature embryo sac. So in the next slide I can show you that a clear image of that embryo sac. So this is a embryo sac structure. So here you can see, so 2 polar nuclei. So nuclei wise 2, but cell wise 1. Here you can see this image clearly, that give 2 nuclei but 1 cell. This one is a egg cell. On either side, that purple color one is that synergy, that orange color one is a synergids, and these 3 are the antipodal cells. Similarly, that sac is surrounded by the 2 layers. You can see some cyan color materials are there. So that is that the outermost one is outer integument, and inner one is inner integuments. So these inner and outer integument meet that this area, some opening also there. So that end is known as a micropylar end. Opposite to micropylar end are the end where that antipodals are there, that is at salagel end. So 2 ends are there, micropylar end and salagel end. So here is a pakka organization of the that embryo sac. Here you can see this is a micropylar region. So these two are synergids, and the middle of that synergids, that egg cells are available. and the whole middle of this embryo sac, that the polar nuclei will be available on the central part. The another opposite end, 3 cells, antipodal cells are arranged. So this is a matured embryo sac and ready for pollinations. I already told you as and when that the anthers are liberated from the pollen grains are liberated from the anther sac, that will reaches the stigma surface of the female organ, where that stigma surface, that pollen grain will germinate and form of pollen tubes. That pollen tubes will this is a stigma area. So here that pollen will germinate and pass that pollen tube through the style. So this entire thing is a stylar region. So that finally reaches that ovary area. So there the ovary, within that ovary the ovules will be there. Now that ovules also in the form of a embryo sac. So as and when that reaches the embryo sac, that pollen tube will penetrate the embryo sac through different mode of entry. Say example, in most of the cases that pollen tube will enter into the embryo sac through the micropolar opening. Here you can see through the micropolar opening and reaches the synergids and finally that pollinate that egg. So this kind of entry, this mode of entry popularly known as porogamy. So that is a entry through the micropolar region. The another mode of entry is entry of the pollen tube through the salagel end. So this end. So this will take that route and through that integument, and that finally reaches the synergids. So this kind of entry is normally known as salagogamy. So here that the pollen tube initial entry is through the salagel regions. Then another thing is, another case is mesogamy. Here that initial entry of the pollen tube either through the funiculus or through the integument area. Funiculus means the point of attachment of the embryo sac with the mother tissues. So through that only that will receive the nutrients. So here that entry is through either integument or that the funiculus. Initial entry, then that will reaches that synergids area, there that will deliver that to male nuclei. So for fertilization process. So for the porogamy, salagogamy and mesogamy, some examples also available. That the porogamy, that is the entry of the pollen tube, that the micropolar region is a very common occurrence. Most number of plants that kind of entry is reported, whereas salagogamy and all reported in the case of casuarina. Whereas mesogamy, particularly the entry through funiculus, is reported in Pistacea, and the entry through integuments are reported in the Cucurbita. So this is a mode of entry or route of entry of the pollen tube into the embryo sac at the time of the fertilizations. So the next one is, so the pollen tube that will carry 3 nuclei. One is that tube nucleus. As and when that reaches that embryo sac, the tube nucleus will be disintegrated. That nucleus is a guiding nuclei for placing of that the pollen tube into the embryo sac. So as and when that reaches that embryo sac, the tube nuclei will disintegrate, whereas the pollen tube that will have 2 sperm cells, 2 male nuclei, that is in haploid nature. So one of the male nuclei that fuse with the egg cell that is located nearer to the micropolar region, on either side of the that egg cells, 2 synergids will be there. So the one of the male nuclei that fuse with the egg cell, that will form a 2n zygote. Later this zygote going to develop into a embryo. So that one egg cell fused with one more nuclei and form a 2n zygote, because the one male haploid cell, one egg cell also haploid nature, so 2 haploids combined together that form a 2n zygote. Whereas another male nuclei, so actually 2 male nuclei, this one going to fuse with the egg, another one male nuclei that will fuse with the 2 polar nuclei that is available in the middle portion of the, here you can see this is the two polar nuclei that is available in the embryo sac. So these two are combined with the one of the male nuclei that form a triploid 3N endosperm nucleus. Triploid endosperm nucleus. Here you can say, you have to remember these points. The endosperms are 3N nature, whereas that zygote and all 2N nature, because in the case of endosperm formation, two polar nuclei, both of them are haploid nature. So two polar nuclei going to combine with the male nuclei. Male nuclei also haploid nature. So three cells are united together. So that process popularly known as a triple fusion. So out of that triple fusion process, the endosperm cells are developed. So here the total process, two type of fertilization is occur. One is that pollination of the male nuclei with the egg cell, that is a one. The second one is that union of the male nuclei with the polar nuclei. So because of that, this process very popularly known as double fertilization, because here the double fertilization occur. As an outcome of the double fertilization process, that zygote and endosperm nucleus were developed. So immediately after fertilization is over, the tissues of the zygote and the tissues of the endosperm that will enter into the mitosis process, that is a multiplication of the cells, and coupled with the organogenesis, that is the tissue modified into various essential parts of the seed system. So here you can see the process of the embryo development in the case of monocot and dicot. The right hand side, whatever that informations are given, that is related to that embryo development in the case of a monocot, whereas that left hand side, the information presented regarding that embryo development in the case of a dicot. First I can explain the matter related to the dicot. So immediately after pollination, the zygote will be in two cell stage, so that will undergo that mitosis, that is a multiplication divisions. So as a result, that will form a 6-cell stage. On the basal part, the whatever that cells are there, that cells are known as a suspensor cells. The top one, that is that area related to that embryo. So again, for the suspensor and proembryo, that will go for several round of multiplication process through mitosis. At the end of the mitosis, that cotyledon will be developed. Cotyledon and primary embryonic axis will be developed. During the several round of replication, the suspensor cell will be fully utilized for the development of the cotyledon. Finally, that suspensor cells also disintegrated and disappeared, and this is a finally developed cotyledon in the case of dicot. So the dicot will have tissues corresponding to that cotyledon, and epicardial region, hypocardial region, and region related to the radicle, and region related to that plumule. That epicardial means that the region above the cotyledon leaf. Hypocardial means the region below the cotyledon leaf. Okay, so I will give you much more clarity about epicardial and hypocardial when while we are discussing about that seed structures. So this is with reference to that embryo development in the case of a dicot, whereas in the case of monocot, there also that follow that same rule, that is that that mitosis process, two cell that undergoes several round of replication that leads to development of the proembryo. So that proembryo that reorganize with reference to formation of that storage organ and embryo. Here you can see clearly. So this is that embryo region. So this is the endosperm area. In the case of embryo regions, so this area is related to that plumular region. So this is related to that radicle. So plumule is related to that shoot development, whereas that radicle is related to that root development. And another peculiar thing as far as that monocot is concerned, so the monocot initially that will have one cotyledon, and that cotyledon will be fully utilized by the developing endosperm. So because of that, the cotyledon is very much reduced, that is available in the monocot seed in between that embryo and endosperm in the name of scutellum. You can see. So in the name of scutellum, that is here. So this is a frequently asked question in most of the examination. What is the name of the reduced form of cartilagen in the case of a monocot? The correct answer is scutellum. So here again, this is a embryo region of the monocot. Actually, it's maize seeds. So this the primula region is well surrounded by sheath-like covering. Here you can see that arrow-like structures are available. So that is that coleoptile. Similarly, that root also surrounded by the arrow kind of things. So that outer covering is known as that coleorhiza. So this is a thing with reference to that embryo development in the case of monocot and dicot. So the next item is endosperm development. So endosperm development normally occur in the case of monocot. So that endosperm, it's a non-living and triploid nature, and it's a storage organ. So here, based on that cell division and organogenesis process, the endosperm development also divided into three types. That is a cellular endosperm, nuclear endosperm, and halobial endosperm. In the case of a cellular endosperm, that cell divisions associated with the that cell wall formation. Here you can see immediately that when that cell division is over, and that the nucleus will be well surrounded by the cell wall. There won't be any free nuclear stage in the case of cellular endosperm type. Whereas this is the nuclear endosperm development. So here that the cell will undergo several round of multiplications, and that nucleus and all available in a free nuclear stage. There won't be any cell wall formation immediately after the that cell divisions. So at the end of that several round of cell multiplication only, that cell wall will be formed. So this is that nuclear type of the endosperm development. Whereas this is a halobial type. So this is an intermediate between the cellular and nuclear. Side by side that division will be take place, that is a multiplication, cell multiplication will be take place, and side by side that cell wall also development also take place. So that is a halobial type. So the halobial type is very common in the case of a monocot, whereas cellular endosperm is development is reported in the case of magnolia and lobelia and avocado, whereas nuclear endosperm development is reported in the case of apple, wheat, and squash. So these are all the things happening with reference to endosperm development in the case of monocot. So now we discussed about that what is happening at the time of the fertilization. So double fertilization, triple fusions, and then embryo development both in the case of monocot and dicot, and endosperm development in the case of monocot. With that idea, I wanted to give you some information regarding that seed maturity also. So through the process, at the end of the pollination fertilizations, the seed structure will be formed. Once that endosperm or cotyledon is formed, automatically the mother plant will start to term the photosynthetes in the embryo, the endosperm, as well as in the cotyledon. In the case of dicot, cotyledon acts as a storage organ, whereas in the case of a monocot, endosperm acts as a storage organ. So this is in the case of a barley. You can see that is a 15th day after flowering. So that is a 15th day after flowering, the mother plant started to term that storage food material, that is dry matter accumulation will initiated, that is 15th day after flowering. So as and when that reaches somewhere around 57 days after flowering, the seed is fully packed with nutrients. So that is a stage, that is the optimum accumulation of the food reserves. After that, after the physiological maturity stage, there won't be any incoming from the mother plant or the seeds. So because of that, physiological maturity means that is a optimum accumulation of food reserves will be take place. Beyond that physiological maturity, there won't be any bioconnection between the mother plant and the seeds. say, and mother plant, due to lack of that bioconnection, the photosynthesis produced by the mother plant also not able to transfer into the seed system. So because of that only, if you are going to retain your seed crop in the field beyond that of physiological maturity, means there won't be any meaning. But the only drawback with reference to that physiological maturity is, you can see that another line, as and when that seed attain that pollination fertilization is over. So at the time of flowering, the moisture content will be somewhere around 85% or something. So that is the 15th day after flowering. Then after that duration is prolonged, the moisture content also started to decline. So here you can see this. So this is a stage at which that optimum accumulation of food reserves is there. So that is that physiological maturity. So in this particular case, during that physiological maturity, the moisture content is 50%. So nearly 50%. So now the problem is the seed attain physiological maturity. So the bioconnection also get lost. The seed also have a 50% moisture content. The seed with the 50% moisture content not getting any protection from the mother plant, means the seed is highly amenable for pest and disease attack. If at all any rain occur, the seed also will germinate because there won't be any support from the mother plant. As long as the seed is get connected with the mother plant, biologically get connected with the mother plant, if at all any rain occur, that won't germinate. Only very few crops like groundnut and green gram, in that case you can see that in situ germination. Whereas the bioconnectivity is available between the mother plant and seed, if at all any rain occur, the seed won't germinate. Suppose that the seed attain physiological maturity, at the time rain going to occur, means whatever that seeds are available in the plant, that will germinate. And also the moisture content also more. So normally in some other crop that will be 17%, that will ranges up to 50% and 55% also. So the moisture content is more than 17%, and the seed coat everything will be soft, so that is amenable for the pest and disease attack. So that is the thing. So suppose you are going to retain your crop until that reaches that harvestable maturity, means you are going to take a risk. Then under that situation, why the farmers they are maintaining the crop to reach that crop harvestable maturity, means the only advantage of maintaining the crop to attain harvestable maturity in the field level, means during that harvestable maturity, definitely that trimeter accumulation is equal to that physiological maturity, or even less than that physiological maturity, because and the day of attainment of that physiological maturity, the seed will initiate that the aging process will be initiated within the seed system. Aging process means degradation of the stored food material for the purpose of running the regular routine biological process. So on the day of physiological maturity, the seed will initiate the aging related process. But the only advantage with reference to harvesting of the crop at harvestable maturity is, here you can see that moisture content is somewhere in between 14%. That is a 14%. This is an ideal moisture. Immediately after harvest, we can go for that threshing, cleaning and grading. And here for making that seed to reach as 14% moisture content, here we are not using any instrument, we are not paying any money. Simply we are keeping the plant, we are exposing the plant to sunlight through natural light, that moisture content is reduced. So without paying anything, we are achieving a desirable moisture content. So immediately after harvest, we can go for that threshing, cleaning and grading. That is the only advantage. But for this, we are taking risk for about 15 days. That is, the seed is attained that 57th day physiological maturity, means after say example 16th day. Then another 15 days we are going to keep that material in the open field conditions. During that period of time, you if at all any rain occur, the seed will germinate. And also if at all any pest and disease that enter in our field, almost all the seeds will be attacked by them. So these are all the drawback. So previously we don't have any instruments for drying for reduction of the moisture content. So because of that, early days the farmers they maintain the crop up to that harvestable maturity. After that they harvested. Now we have a well sophisticated instrument for drying process, for drying purpose. So with the help of that we can harvest the crop at physiological maturity, then we can go for moisture reduction, and safely we can remove the seed from the mother plant. So if you do so, in the quality of the seed will be good. There won't be any question of pest and disease occurrence or that free germinated seeds. Okay, so these are all the advantages with reference to harvesting of the crop at physiological maturity. So these are all the things with reference to that maturation related event in the case of seeds. With that I am completing that seed development and maturation things. So the next class we can discuss about the biochemical process related to that seed germinations. Thank you. So this is the overall development of the what we were discussed so far.